Hello everyone, welcome to our channel. Before understanding chromosomal disorder, we must understand how a normal karyotype looks like or what is a normal karyotype. So, in a normal human somatic cell which contain 46 chromosomes, 22 of them are homologous pair of autosomes and 2 sex chromosomes. In female 2 X chromosome and in male 1 X and 1 Y chromosomes. So karyotyping is the study of chromosome, and karyotype is a diagrammatic representation of the chromosome according to their size, and they lastly followed by the sex chromosome. So how we achieve this karyotype? We arrest the dividing cell in metaphase by using some mitotic spindle inhibitors like colchicine, and then we stain the cell. Metaphase chromosome, as you know, we arrest the cell in metaphase. Metaphase chromosome has 2 chromatids connected with a centromere. This is the centromere, and we have 2 chromatids. This is the metaphase chromosome. So in karyotype, arranging each pair of autosome according to the length. First chromosome is the largest and 22nd is the smallest, and autosome followed by the sex chromosome. So this is a normal karyotype of a male, in which we have chromosome 1, 2, 3, 4, 5, 6 till the chromosome 22, and then followed by the sex chromosome. So these chromosomes arranged in a order according to the size. What is G bending? Giemsa stain is used for the staining of these metaphase chromosome, and in this Giemsa bending there is alternating light and dark bands are there. That's why this is known as G bending. Total 400 to 800 bands per haploid is seen in the G bending karyotype. Now we should understand certain cytogenetic terminology before understanding the chromosomal disorder. In a karyotype, first we write the total number of the chromosome, then we write sex chromosome. After the sex chromosome, we write description of anomalies in ascending order. For example, in a normal male karyotype, 46 is the total number of chromosome followed by the sex chromosome, which is X and Y. In a normal female karyotype, 46 is the total number of chromosome and followed by the 2X chromosome, 2X sex chromosome. Likewise, a female with trisomy 18, denoted as total number of chromosome, which is 47, followed by the sex chromosome, 2X chromosome, and the anomaly, which is an additional chromosome 18. Short arm of the chromosome denoted by the small p, and long arm of the chromosome denoted by the q. So this is some metacentric chromosome in which short arm is denoted by p and long arm is denoted by q. These arms having multiple regions, like we have indicated 1, 2, 3. There are more than 3 regions. There can be more than 3 regions. In every region there are bands, and every band there are some certain sub-bands. So in chromosome we have short and long arm, particularly in acrocentric and submetacentric chromosome. Arms are divided into regions are divided into bands, and bands are divided into the sub-bands. So this XP21.2 represent a chromosomal segment located on short arm of, we denote the short arm of X chromosome in the region 2, band 1 and subband 2. So this is how it denotes a chromosomal segment. We should know this terminology to precisely locate a gene or locus. What are the structural abnormalities of a chromosome? So the structural abnormalities of chromosome, we will discuss in very brief about these. Aberration of underlying cytogenetic disorder can be due to the abnormal number of the chromosome or the alteration in the structure of chromosome. So these are the two main reasons for the cytogenetic aberration or cytogenetic disorders. Abnormal number of chromosome can be less or more, or alteration in the structure of the chromosome. Normal chromosome complement expressed as 46XY in male, 46XX in female. What is euploid? Euploid is any exact multiple of haploid, which means 23 of the chromosome. Any exact multiple of the haploid number of chromosome, like 23, 46, 69. And what is aneuploidy? Not an exact multiple of 23. And this is because of the error in meiosis and mitosis. For example, trisomy 21, in which the total number of chromosomes are 47, not exact multiple of the 23. The usual cause of aneuploidy are non-disjunction or anaphase lag. There are certain terminology like monosomy and trisomy. Monosomy is represent loss of one chromosome, and trisomy represent gain of one chromosome. So what is non-disjunction? As we are discussing, aneuploidy are because of the non-disjunction or anaphase lag. In non-disjunction, there are error in the cell division. Meiosis can be meiosis or mitosis. Homologous chromosome or sister chromatids fail to separate properly, and this may result into the one daughter cell receive one extra chromosome and other daughter cell lacks one chromosome. So when a one daughter cell receive one extra chromosome, then this condition is known as trisomy, which is seen in, for example, in Down syndrome there is trisomy 21, one extra 21 chromosome. And when a daughter cell receive one chromosome less, then this condition is known as monosomy. What is mosaic or mosaicism? Two or more genetically different cell lines within the same individual from a single fertilized egg. Two or more genetically different cell line within a same individual from single fertilized egg. Mutation of or chromosomal changes during the early embryonic cell divisions are predominantly responsible for the mosaicism. In the individual with mosaic, there is a mixture of normal and abnormal cell can be seen. Translocation. A segment of chromosome breaks and attaches to the another chromosome. In translocation can be balanced when there is no genetic material lost or gained, or it can be unbalanced in which an extra or missing genetic material can be seen. So in this case we can see the translocation of these material to each other. And one of the subtypes of translocation is robertsonian translocation. Translocation in two acrocentric chromosome, in which long arms of two acrocentric chromosome fuse at the centromere. So these long arms of these two acrocentric chromosome fuse at the centromere, and the small arms along with the small part of centromere lost. This is seen in familial Down syndrome. Now, what is deletion? Loss of a segment of a chromosome. A segment of chromosome is lost in deletion. For example, deletion 5p, a small part of short arm of chromosome 5 is deleted, which is seen in Karyotype syndrome. What is an isochromosome? Abnormal chromosome with identical arms. Centromere divides incorrectly in the formation of isochromosome. Two copies of either short arm or long arms are there. So example of isochromosome is isochromosome XQ, which is responsible for the Turner syndrome variant. Ring chromosome. This is a special form of deletion in which when break occurs both end of a chromosome with a fusion of the damaged end. Like in this example, breaks occur at the end and then followed by the fusion and formation of the ring chromosome. Now we will discuss about the inversion. So what is inversion? Two breaks within a single chromosome with incorporation of inverted intervening segment. Two breaks within a single chromosome with incorporation of the inverted intervening segment. So inversions are two types, paracentric and pericentric. In paracentric inversion, only one arm of the chromosome is involved, like this segment of chromosome. This segment of chromosome is inverted. And in paracentric inversion, opposite side of the centromere is involved, like this segment is inverted in pericentric inversion. So this is basic about the chromosomal disorder and normal karyotype and various terminology we are going to use in the chromosomal disorders. Now briefly we will discuss about the types of chromosomes. Types of chromosome based on the position of the centromere. When the centromere is in center and both arms are equal, this is known as metacentric chromosomes. In submetacentric chromosome, the centromere is placed slightly one side, in which short arm is slightly shorter than the large arm or long arm. Most of the human chromosomes are submetacentric chromosomes. Acrocentric chromosomes, in which short arm, there are a small short arm and a very large long arm. Bichromosome is an example of acrocentric chromosome. Telocentric chromosome, centromeres are present at the end. There are only two arms. No short arm is there. This is rarely seen in the human. So on the basis of the position of centromere, 4 types of chromosomes are there: metacentric, submetacentric, acrocentric and telocentric. Thank you for your time.